Good evening, everybody. Thank you very much for coming for Café Scientifique once again. And if you are coming for the first time, welcome, everybody. Just going to let them come in. Please join, sit wherever is available. Just before we start, can we have a show of hands, please? Those of you who are not in Homestead. Thank you very much, and thanks once again for coming. Today we are going to have Dr. Adriana Torres-Ballesteros. She's a microbiologist, microbiological ecologist, and data scientist at Rockwood Research, specializing in soil microbiology and sustainable agriculture. She's a scientific director and co-founder of Mind and Microbes, an organization dedicated to advancing microbiological science communication. Her research explores the role of microbes in mitigating climate change, with a particular focus on integrating microbial ecology and physics. Currently, she's leading efforts to establish the Biosphere and Climate Resilience Research Network, fostering interdisciplinary and international collaboration in this field. Adriana is also an IPCC scholar, contributing to global climate assessment and policy relevant research. So thank you very much, Adriana, for agreeing to give us a talk. And welcome, everybody. Thank you.
Thank you, Anu. Thank you, Claudia and Annalisa, for inviting me. I'm very happy to be here today because today is officially the Women's in Science International Day. So I'm very happy to be here presenting some of the work in science that I have been doing. And I'm going to talk about two different stories. One is tropical methane, and the other one is signals in the soil. Tropical methane is related to my work in Brazil when I was doing my PhD, and signals in the soil is the work I did at Harpenden. So I'm going to start with the tropical methane. And this was very interesting because the main source of electricity in South America is hydropower reservoirs. And they sell this source of energy as clean. But when we have a reservoir built in Brazil, this is what happens. You cut a lot of trees and then you flood the area to have a reservoir. Sometimes they don't cut the trees, so the trees are still there, but are flooded. So that will change all the dynamics in terms of biology and microbiology especially, and the dynamics of greenhouse gas emissions. So what happens with a reservoir is like there is a lot of organic matter, then comes the water, and that's the perfect space for one kind of microbes to grow. This kind of microbes produce methane. So these microbes are called methanogens, and they do methanogenesis. But there are also other kind of microbes that use this methane as an energy source. So there is a balance between these two kinds of microbes, the ones that produce methane and the ones that consume methane. And at the end, you will have an emission that is the result of this balance between the two communities. When we talked about greenhouse gas emissions, carbon dioxide is the most emitted gas as a result of anthropogenic activities compared to methane. But if we look into the global warming potential, methane has 25 more capacity to capture heat in the atmosphere, so has a larger global warming potential. So this looking into this problem, we designed a sampling across Brazil, different areas, and we selected 8 reservoirs. Here in this site, where is it? Okay, on that side, we see one of the examples of the sampling points in one of the reservoirs of the 8 that we sampled. Then, on each of these sampling points, we use these floating chambers and waited for different times to get the samples for different gases, and then we took these gases to the lab to measure the quantities of methane and carbon dioxide. But we were also interested in what were the microbes doing there and how they were shaping the emissions of greenhouse gases and especially methane in these reservoirs. So we took samples from sediment and water and target 2 different genes from these communities. One gene for the ones that consume the methane and another gene for the ones that produce the methane. And in each point, we found that doing some calculations, if we balance the number of microbial communities that consume and produce, we can find different trends in the hydropower reservoirs that we sampled. So this is one of the examples in these reservoirs that are closer to the Amazon. The dynamics show that the relation between the communities that produce and consumed the methane were proportional. So that was what we were expecting. But then and the same for the reservoirs that were more in the middle of Brazil. That was different for, where is that? Yeah. It was different from the reservoirs that were in the south of Brazil. Okay. So the main things we found is that these communities were positively correlated to the methane emissions. So if we can manage the numbers of the communities that can consume the methane, we can reduce the impact of hydropower reservoirs in Brazil, mainly in the north. Then for the south of Brazil, we need to think about, in how we regulate another kind of environmental factors that will affect the communities that consume the methane. For example, increasing oxygen and reducing the times that we store the water in the reservoir. This work was sponsored by the International Panel of Climate Change. I got a scholarship from the Prince Albert of Monaco, and then is when I started to work with to find interested in how microbes can shape climate change, not just in reservoirs but in different environments. The other story, signals in the soil. This was a project in partnership with different institutions in the States, with the Colorado School of Mines and Berkeley University. The idea was to develop new sensors to measure CO2. Instead of taking the samples from the chambers and take it to the lab, the idea was to use fiber optics in the soil to measure different concentrations of CO2. This is one of the prototypes that they developed, but it's still in progress. So to that, to work in this project, we used one of the long-term experiments. So Andy Gregory was here last time. He talked about this. So we used this long-term experiment and installed these fiber optics and different probes to measure the CO2 dynamics in soil. This one was one of the probes that I used there. We just buried this into the soil at different depths and then used this machine to get the concentrations of CO2, methane, N2O, and the main greenhouse gases. And why did we use a high field? Because this experiment, we know that the different treatments present a different soil structure. For example, a treatment that is grassland has larger pores, and the pores are more connected compared to bare fallow and arable treatments. Here is a 3D view of an aggregate comparing bare fallow and grassland, and the gold spots are representing the pores. So you see in grassland there is more connections and more pores compared to bare fallow. We thought that that could be an interesting factor that can impact the differences in CO2 concentrations in the soil. So this was the plan: install the probes at 3 different depths, because in the different depths there are different kind, we believe there is different kind of microbes and they have different functions. And then we used this equipment and we collected data for 7 months using the high field experiment that has these three different treatments: grassland, arable, and bare fallow. And physicists were very happy to see a correlation between soil moisture that is in this x-axis with the CO2 concentrations, but that was easier to see for arable and bare fallow. For grassland, it was more difficult to explain just looking into soil moisture. So that's why microbiologists came to the project. Oops. Sorry. And then doing this, using a similar approach that we used for the project in Brazil, but targeting different genes, we found that, as well as the results from the pores, the treatments of arable and bare fallow were more similar when compared to grassland. We look at 3 different groups of microbes: bacteria, fungi, and protists. And then if we look into the bacteria, there is not much difference. But if we look into protists, when we can see that there is a big difference in the number of shared microbes between arable and bare fallow and grassland. So these are, so what we want to do now is to continue to use the gas probes and get data from different fields to use in a model that helps us to predict what will be the emissions from different kinds of soil and land management, and then use some techniques to capture this CO2 in soil. So this is the 2 stories I wanted to talk to you about. It's just a summary of 2 works that I have done looking into microbes and climate change. We have this foundation, Mind the Microbes, and we share a lot of information related to how we can use microbes to mitigate climate change and also clean soils and water. So I want to acknowledge all the co-workers and advisors and all the institutions that were involved in signals in the soil. And if you want to have a look in how this machine works, then we can see it a little bit. Thank you.
You want to demonstrate the machine now, or
No, it's okay.
can you just come?
Yeah, just come. Yeah, just come. It's easier. Yeah.
Thank you, Adriana. So do we have any questions? Questions?
If there's anything you want to go into more detail about as well,
Yeah.
good. Like, or go back through some of the slides, like potentially, like, because I missed the first part of it as well. So, looking at your microbial communities in the reservoirs, what would you say were the more contributing factors towards the differences in there? Because I imagine, like, it was hard to get a good sort of set of replication of similar conditions in there, so that was quite hard to do.
The organic matter, the content of organic matter will promote the growth of the communities that produce methane.
Okay.
So if you cut the trees before doing the flooding, then you will have less methane emissions in the future. Comparing when you have more organic matter, for example, when you didn't cut the trees.
Okay, yeah. So I'm guessing you're saying like a management system would need to be in place to ensure that methane conditions go down.
Also, the retention times in the reservoir, if you are you change that with time, like shorter time are better because they will increase oxygen in the water. So then the communities that consume the methane will grow better. Okay. Then they will have less emissions into the atmosphere.
And what about potentially like the size of the reservoir? Because I'm guessing if you have like smaller reservoirs, they're going to heat up more quickly than large ones. So again, temperature could be a contributing factor.
Yeah, that's why in the south we had a different pattern, because in the south the size of the reservoirs was smaller, and also the temperature in the south is they are cooler.
Oh, okay.
Yeah.
So they're cooler even though they're smaller?
Yeah.
How?
Because of the climate.
Oh, because of the climate, okay.
Because of the region, yeah.
Yes, okay, yeah, okay. This is what happens when you come in a bit late.
I'm assuming that the amount that it's mixing would also have an effect. Did you find any correlation with depth, so that something that was a deeper reservoir had less flow through and it was mixing less, or was there any associations there?
Yeah, like deeper was better because also there was more abundance of communities that consume the methane. So deeper was better because there was more space for the communities that consume to grow.
And presumably all the reservoirs are roughly the same age?
Different age.
Was there a correlation? Because presumably if you've flooded ground for longer, maybe 100 years, your methane producers will die off because the organic matter at the bottom of the reservoir will drop, and therefore the longer the reservoir is in existence, less methane will produce. So therefore, hopefully, in due course, the methane emissions from these reservoirs will drop off.
Yeah, so the age of the reservoir will have an impact on the measurements. We did it in the 2 years and then compare different ages. Then, at the beginning will be a peak of methane and then will be stable for a long time. Yeah.
Yes, please.
So, what was your overall conclusion about how clean the hydropower was compared to other things like fossil fuel power generation or whatever? How did it compare?
No, clean. It's not clean.
Really?
Yeah, yeah. So they try to sell it as a clean, but if you do like a carbon cycle assessment, then you will see that it is almost equal to carbon coal.
Wow.
Coal. Wow.
Wow. That's an important result.
Yeah, and that's not the only issue. There is also the issue of impacting the indigenous communities that lives around the river.
Yeah.
So there is not just impact in terms of greenhouse gases, but all the communities, the fish. Yeah. So So
do you think that conclusion would be true around the world for hydropower?
No. No. As the examples I showed in the north is very different from the south. The trains just change. So I think we can't extrapolate that. Yeah. I think for Europe it has less impact than South America. Also because some of the reservoirs were built near the Amazon or in the Amazon. Yeah.
So is the temperature of the water, you think, is the primary correlation in terms of how much methane is being produced?
Can you repeat, please?
Is it the temperature of the water that's having the most effect on the amount of methane that's being produced?
It's more the carbon, the organic matter. Yes. Yeah, it's more that.
You had a question? Yeah, a couple of questions. So is it you're saying it's as bad as coal? Is it as bad as coal both in the north and the south, or is it just as bad as coal in the south but the north is okay?
The north is bad, the north of Brazil. Yeah. The south you can balance.
Okay.
Yeah.
So are there moves by the government or whoever controls electricity production to change and to move hydro generation to the south, I guess?
Yeah.
Or not build any more in the north?
Yeah, at least that, not built anymore in the north. Yeah, yeah, there was a lot of political movement around that. Yeah, so it is very in South America it's very conflictive to publish this data because there is a lot of political interest, because they were selling this as clean and it was part of a political program. Then if we present this then it will be
they've taken it on board?
not the past president, not Bolsonaro, but Lula is open to hear the results about science.
Have similar studies to the reservoir one been done in other countries? Because also China is very like hydropower fan sort of thinking. So was there any other studies to compare with?
Yes, in China, but the thing is, in those countries there is a more diversified network, so you can compensate the impact of hydropower with other cleaner energies. Yeah. But in South America, that's the main thing. Okay. And when there is no rain, there is no light in the city, the main cities. So it's a big issue.
Yeah. I can see now why they were promoting them. They are as bad as coal.
Yeah.
Hi, Adriana.
Hello.
First part of your work at Rothamsted focused mainly on looking at long-term grass, long-term arable in that project. I didn't know if you wanted to say a bit about what you plan to do next, if it's more work on those experiments, those treatments, or if you're looking at other experimental treatments.
I want to look into the long-term experiment, mainly high field. What I want to look is specifically to the functions at different levels. These are just preliminary results, but I want to know how the different communities function different depending on the soil structure and how they are different depending on the size of the pores. That's one of the things that I'm interested in, but I'm working with other projects with agroforestry now, looking into different ways to do farming, and then it's good to have these two long-term data and then get these new farm practices and compare how the microbial dynamics change and how that will be reflected into the greenhouse gas emissions. Yes.
When you were taking samples and then in the reservoir, how much spatial variation was there between different parts of the reservoir? And did it depend on any particular factors, like where there was originally a river valley at the bottom of the reservoir, or where there was a ridge?
We selected depending on how close it was to the dam, how close was to the river, and then select different kind of replicate of the number of sampling in each of these regions for all the reservoirs, so we can make it a little bit comparable. But that was the main thing, just how close we were to the dam and how close we were to the river. Yeah.
Do you want to carry on with more questions?
Yes.
Okay. I do feel we're grilling you a bit. So I was thinking back to essentially your very, it seems to me, high-end technology that you now have to do all these measurements with the Rothamsted one. So I was thinking back to your really cool 3D rotating pictures to get your pore density slide in there. And I was trying to think back to my undergraduate days when I did some soil sampling, and essentially to do pore density, I think you just had some sort of 2D images and a ruler and trying to calculate spaces between there, So I was my overarching question is how much could you have done this type of research you wanted to do even just 5 years ago, or has the technology to do what you're wanting to look at exponentially sort of manifested, or is it more of the technology has always been there, it's just looking at the applications you can use it for in agriculture? It's how important is this technology to this work that you're doing right now?
So for the reservoirs, I did almost 8 years ago.
Okay, yeah.
So we're still using the same tools, of course. Every year we have updates.
Oh, yeah.
But to look into the microbial communities, we use mostly sequencing, and the baseline is the same.
Yeah.
But there is always room for improvement. So I think from 8 years ago to today, we still use the same kind of tools. In terms of the soil structure also, there has been a lot of improvements, but this technology existed many years ago. Yeah.
We have another
If
question.
you've got time for one, can you explain a bit more about the fibre optic that you mentioned?
Yes. So the idea is to use a coat into the fiber optic, and then the CO2 reacts to that chemical that is around the fiber optic, and then the signal of this reaction will be detected using the fiber optic. This is connected to a machine that will detect those changes in frequency. So there is a source of light that travels into the fiber optic, and this light will change if there is a reaction as a result of the CO2 interaction with the chemical that is around the fiber optic. that's the plan. I mean, it's working, but we still need to work on it. Yeah. So instead of having these measurements in each point of a field, then we will have a lot of measurements along the fiber optic. So it will be easier to make models and correlate, not just using the sampling point, but having a lot of data along along the fiber optic.
Does the technology enable you to detect where the CO2 is?
Yes.
Along the cable?
Yes. Yes. We installed this in Highfield. You can go and see the hole that we did to install the fiber optics. But we also had this at Woburn. We installed 300 meters of cable there, not to measure CO2, but to measure the cracks that that are formed in the soil. So then we can detect where the crack is along the 300 meters of cable.
But if it's just a light signal, how do you know where that emanated from? When there is for example, for the cracks, if we stretch in one section of the cable, then the light will have a less signal. So then we can detect along at different points. Yes? Yeah. Thank you.
Any more questions?
Are you able to say a bit about how your work in the soil at Rothamsted might impact farming policy?
I would like to have a big impact in the terms of how soil is managed and which practice can help us to capture more CO2. And then how can we keep our soil health in not just in UK, but in different countries. But yeah, that will be the main thing. In which practices can we use to reduce the CO2 emissions and capture more CO2 and store CO2 in the soil? That will be, yeah.
I think the caveat there is the extrapolation, like with your reservoirs. The results you get there don't necessarily extrapolate. I would hesitate in extrapolating the data you get from just up here with other soils and other climates. And it could be quite dangerous if you did.
Yeah.
I think Highfield is a fascinating place. I could spend weeks and weeks up there. And the stuff you're doing is fascinating. With the big thing is that the structure between the difference between the grassland, the arable, and the fallow. But is there a particular time when you do that soil thing? Because I was a bit surprised that when I went there last, the way you control the vegetation on the bare soil is by ploughing it, which in my book says you're introducing a whole stack of air, or you're disturbing a whole stack of microporosity that would have been there, perhaps might have been there otherwise. So there's a little bit of artificiality in there that I don't know how you're taking steps to reduce that.
As is a long-term experiment, we have like when the plants grow there and then plowed is a shorter time compared with all the time that we have planted the arable plots and also all the time that grassland has been there. So I think it's because of the long-term characteristic of the experiment, then we can reduce a little bit of the impact of this plowing.
Yeah, I think. Yeah, took.
So that we also have other experiments where we're no longer ploughing. So other treatments where we're trying to reduce that impact. We also have, so there's new long-term experiments starting not that far ago, not that long ago, but about 2018, but where we have different, sort of non-till versus till, cover crops versus non-cover crops. So we're trying to incorporate, all these type of things into new experiments. So as Adriana was saying, this one is an old experiment, and in those days people didn't think about things like that, but that's something that we're sort of trying to incorporate into the new experiment.
I can see it can be quite difficult extrapolating this, but what's the time frame for getting results for how it plays out in Harpenden, in Highfield, in this area? Is this a 2-year, 5-year, 10-year experiment, or do you expect to get results?
When I do expect to have results for
results for out of the areas that you've got in Highfield, which are the best environments, or which is the best planting schedule?
Yeah.
Per the experiments that are in that are.
Yeah, that will take some years because that's the idea of having a network of farmers or a networks of different countries, so we can have data from different places, and try not just to extrapolate, but having.
I was wondering how long it will take for just which of the Harpenden environments are better, so the arable versus grassland, of the ones within the area you've got here. Is that, do you know that already, or is that something that's going to
we have some preliminary data, but I think in another 2 years, then we have more.
Okay. So we might hear in 2 years' time which of these are the update. Yeah.
Have you got any plans to do any of this work on things like no-till and cover crops in the future? Or?
Yeah, as Ian said, there is a lot of new experiments, and there's plans, but also we need funding.
There's always plans.
Yeah, we need funding and people to work. Because there is a lot of time looking into this, but that's why working in models that will reduce some of the time.
Yes.
Yeah.
So, thank you very much, Adriana, for the wonderful talk and the wonderful interactive Q&A session to everyone.
And the gizmos.
And would you like people to come and talk to you?
Yes. I just want to have this conversation instead of giving a talk. But if you want to come and see how this works, then you are welcome.
People will hang around here, so feel free to have. And so thank you once again, and let's give a round of applause for Adriana.